This is Lecture 8 from Unit 1, the final lecture in this unit. In this section we are going to be looking at cell division. Somatic cell division is the division of non-sex cells, which are all of your cells except the sperm and egg. Interphase is where the cell spends most of its life cycle. This is the non-dividing phase where the cell is doing its normal metabolic activities. During mitotic phase, it is the actual dividing part of the life cycle. Mitosis is the process of cell division and has 4 phases. During prophase, the chromatin forms chromosomes in the nucleus. You will also see the start of the forming of the mitotic spindle, which is used to pull the cell apart. The nucleolus will also disappear, and as you can see in the picture, the nuclear envelope will start to disintegrate. In metaphase, the chromosome that formed earlier begin to align along the center. The nuclear envelope has completely disappeared at this point, and the mitotic spindle begins to elongate, preparing to stretch the cell into 2. In anaphase, the sister chromatids begin to separate, so the chromosomes are pulling apart and forming 2 separate halves. You have cytokinesis here, which is the division of the cytoplasm. The organelles will split into the 2 sides of the cell. There is a cleavage furrow forming where the membrane around the cell is going to pinch to form 2 separate cells. In telophase, for the most part, our separation is complete. The chromatids go back to being chromatin, a new nucleolus will form in the nucleus, and the mitotic spindle disappears. You will also form a new nuclear membrane. Here we will watch a link showing the whole process of mitosis tied together.
Mitosis is a process of nuclear division in which the duplicated chromosomes separate to form 2 genetically identical daughter nuclei. During prophase, the nuclear membrane disintegrates and the nucleolus disappears. As prophase continues, the chromosomes condense and begin to appear. Mitotic spindles, which are made of microtubules, begin to form between the poles, and kinetochores begin to mature and attach to the spindles. During metaphase, the chromosomes become attached to the mitotic spindles at the kinetochores and align along the metaphase plate at the equator of the cell. During anaphase, the kinetochore microtubules shorten, separating the chromosomes to opposite poles while the polar microtubules and the cell elongate. During telophase, chromosomes reach the poles of the cell and begin to disappear. The polar microtubules continue to elongate. The nuclear membranes form, the nucleoli appear, and the chromosomes decondense. Telophase is followed by cytokinesis, during which a cleavage furrow forms in the center of the cell, and the cell divides.
Meiosis is the process of sex cell division. We will go into the details of this division at the end of the course in the reproductive system. Just a quick basic overview, the cells undergo a second division without replicating the DNA. This will allow the cells to have half the genetic information as is seen in the sperm and the egg.